Hello, in this video we will discuss about the gene therapy. So what is the gene therapy? In this video we will just look at the overview. So insertion of genetic materials into human cell or animal cell or plant for the treatment of a disorder is known as gene therapy because the defective gene removed to add the other gene which that is non-defective and the beneficial. So the defective gene containing patient is here, which that contains cell and cell contain nucleus contain genome is made up of DNA and histone protein. So this is the DNA is the defective gene containing DNA which that cause the disease. For example, the overexpression of the gene or suppression of the gene. So this is the nucleus, plasma membrane, and the cytoplasm. So gene therapy is two type: ex vivo gene therapy and in vivo gene therapy. Ex vivo gene therapy, first of all, we will learn. And in this way, the ex vivo means the external cell will remove from the tissue of the person, which that will introduce the retrovirus, which that is the famous ex vivo gene therapy. In this way, here is the defective gene, while the normal gene containing retrovirus, which that introduced to the cell, and after the introduction to the cell, the reverse transcriptase enzyme which that present in the retrovirus and the basically used for the recombination of the DNA through a integrase enzyme. So in this way, the SCID, this disease can be recovered by this ex vivo gene therapy. While the in vivo gene therapy, let's begin to understand. In vivo mean the gene, the DNA segment containing gene which that will be introduced inside of the body. So intracellularly, introduction of healthy gene to clear the disorder. It can be hemophilia, diabetes, Parkinson disease, and AIDS, acquired immunodeficiency syndrome. And the normal clotting factor gene containing normal clotting factor will introduce for hemophilia recovery, while the dopamine producing genes could be planted or introduced for the Parkinson disease recovery. So let's begin to understand the person which does contain a bone marrow. Let's begin to exemplify here. So this is the bone which that is basically contain a bone marrow and the compact bone and spongy bone as well as the bone marrow. Bone marrow is containing the pluripotent stem cell which that is able to produce many type of cell, for example B cell, natural killer cell, macrophage, monocyte. There is a lot of things. So containing stem cell is ability to produce and differentiate and proliferate. So the stem cell itself is also dividable and it mitotically divide. So this stem cell which that is introduced the normal gene containing recombinant RNA via a retrovirus. This retrovirus will enter via a different function, different process we will not understand, but here when introduced, so the insertion into defective gene containing stem cell, because the stem cell is basically first of all we should need to understand the stem cell able to divide but also the tissue containing proliferation, differentiate into many cell like B cell, T cell, natural killer cell, WBC and RBC. So that is why the stem cell is a major target for recovery and the healthy gene introduction, because it can be dividable and long time it can be divide more to stem cell will non-defective due to the normal gene introduction. So this is the mechanism. The reverse transcriptase enzyme will begin to reverse transcription of RNA into DNA and the DNA segment recombinant. This recombinant DNA, which that is ligase enzyme molecular paste, which that will be create the sticky end, and the integrase enzyme, which that is a part of retrovirus, will lead to integration into the genome of the stem cell into the nucleus. So after this, basically the engineered cell, the return the genetically engineered cell to the patient bone marrow inside. After the introduction into the bone marrow, it will be proliferate and differentiate into the stem cell to into the B cell, T cell, natural killer cell, RBC, WBC. In this way, RBC if defective or WBC defective, it can be recovered. So the major disease which can be recovered through a gene therapy is the hypercholesteremia, when liver cell lack cholesterol removing receptor in their cell. While the cystic fibrosis patient, which that is basically the liposome, microscopic vesicle containing transmembrane gene containing liposome, which that will introduce for the cystic fibrosis patient, because the chloride ion cannot move for the cystic fibrosis patient, cannot regulate. In this way, the chloride ion transporter protein containing gene will be introduced. While in the last one is the arteriosclerosis is a major example. In this way, the angioplasty, which that is the containing camera containing balloon, which that will and coated with the endothelial plasmid growth factor containing plasmid, which that will be insert into the endothelial cell for the normal function. It contains a basically growth factor, so that is why it will be recovered. Because the growth factor will be defect will lead to arteriosclerosis. Anyhow, let's begin to understand a little bit about the scope of the gene therapy. So the scope of the gene therapy is the future advanced technique used for gene editing before birth to healthy reproduces and diseases free animals and human being as well as the plant. So that is why the scope of the gene therapy is so important, and the next decade it will be revolutionized to gene therapy will maintain and decrease the aging as well as increase the aging and immortality, touching to the immortality of the human being. So this was the gene therapy video about the function of the gene therapy and what is the gene therapy. So thanks for watching. Please make sure to subscribe, like and share. Ask the question in the comment below for making a new video as a suggestion. I hope you make sense about this gene therapy video. So take care, bye.